And so tonight, without any further ado, I'd like to welcome, Peter Sharpe. Peter Sharpe from the Institute of Wildlife Studies will be discussing the return of the bald eagles to their historic range here on the Channel Islands. Peter received his PhD in zoology from Colorado State University in 1998. And since 1997, he has been directing the bald eagle reintroduction program here on the Channel Islands. So please welcome Peter Sharpe.
Thank you very much. Can everyone hear me? Yes. Well, tonight I'd like to talk about a project that we've been conducting since 1980 to bring bald eagles back to the Channel Islands, and why I'm considering it successful restoration. So I'd like to give you a brief overview of the history of the eagles, what we've done, and then an update on the current status of the birds. Now, bald eagles were once found on all 8 of the California Channel Islands, as well as along the mainland coastline. Those along the mainland disappeared probably by the early 1900s as humans moved into the areas, but they persisted on the islands until at least the 1950s. Now, bald eagles had a bad rap. They were considered dangers to people, children, your livestock. They'd come in and carry your babies off. So they had a bad rap, and they were severely persecuted by people. Here they are. They'll just attack you for no reason whatsoever. So people would come out to the Channel Islands. A lot of the islands had things to hunt, like goats. And when they had an opportunity, they'd also shoot a bald eagle. So human persecution probably had some impact on them. But what really drove them to extirpation on the islands was the introduction of DDT into the Southern California marine system. DDT was developed during World War II, used extensively in the Pacific to fight insects. Then after the war, it was brought back to the U.S. and sprayed a lot. Yeah, that's pretty gross. So a lot of you probably saw this being sprayed around your neighborhood. It was also available in many different forms that you could use in your own home to control just about any kind of living organism there was. DDT itself is poisonous, but what really affects the birds is DDE, which is one of the things that it breaks down into. And that impacts how the females lay down the eggshell, and the eggs tend to break before the chick can hatch. So the last known successful hatching on the Channel Islands was in 1950 on Santa Rosa. DDT was banned for use in the U.S. in 1972, and by the late 70s, other birds which had been impacted, such as the peregrine falcons, were again breeding. So my boss, Dave Garcelon, decided to try to reintroduce bald eagles to the Channel Islands. He chose Santa Catalina Island for a variety of reasons. One is the Catalina Island Conservancy was willing to have us do the work out there. It's got a good ferry system going from Long Beach, so there's daily ferries, easy island to get to, probably the easiest island to get to. And the hope was that we'd get a healthy population going on Catalina that would then expand out to the other Channel Islands and maybe any mainland areas which were appropriate. Because there weren't any eagles left in Southern California, we had to travel to places like Northern California, Washington, British Columbia, and find nests with approximately 7 to 8 week old eagle chicks like this in them. What we would do is climb up into the nest, remove one of the chicks, and leave one for the adults to continue raising. We'd then bring them down to Catalina Island and put them into what we call a hacking tower. This is about an 8 by 8 by 8 foot cage up on stilts, so the eagles are protected. We have little doors that we can throw food into them. They don't see humans except for when we go in to ban them when they're about 11 to 12 weeks old. So they're in there, they see the environment, they get the wind, they have room to exercise. So they're in there for about 4 weeks, feeding themselves essentially. So we'd monitor these daily, making sure that each of the birds was eating. If there seemed to be a problem, we could remove it from the tower, but pretty much all the birds did well. Then when they're about 12 weeks old, we would open the front doors on the tower And when the birds were comfortable, they could then exit the cage, get a little bit more exercise, a little more freedom. Eventually, they'll get the guts to fly all the way up to the roof. This is really big time for them. And then eventually, they'll get the guts to actually take their first real flight. Because adults typically feed the chicks, the fledglings, for about a month after they start flying, we would put food in and around the towers so that they'd have a known food source that they could come back to. They'd start exploring the island, but they had a known place that they could come back for food. And eventually they go out and find their own food. So we released 33 eagles on Catalina between 1980 and 1986. By the late 80s, we had some pair formation, and we had our first eggs laid in 1987 and 88. Unfortunately, all these eggs broke in the nest. We were able to get into the nests and collect some of the remains of the eggs and have them analyzed. And just for your information, about 3 to 5 parts per million of DDE in an egg is where you expect to get some eggshell thinning. If there's more than about 15 parts per million, you expect complete failure, just the eggs will break. So other areas that were considered contaminated, such as in the Great Lakes and along the Columbia River, had 2.8 and 9.7 parts per million. So that was considered pretty contaminated. When we looked at our eggs, they actually had almost 33 parts per million of DDE in them. So it was obvious that there was something going on in the area. In the mid-'80s, it was discovered that Montrose Chemical Corporation in Torrance, a major DDT manufacturer, had been discharging DDT through the sewer systems coming off of Palos Verdes Peninsula there. Estimated that over 1,800 metric tons were discharged from 47 to 91. Then they'd also take out barrels into deeper water sites and dump out there. And you can see, how close that is to Catalina Island. So at this point, it became pretty obvious that the eggs weren't going to hatch anytime soon on their own. So we started a manipulation in which every spring, usually actually in February, we'd start watching the birds, find out where they're going to nest and when they lay eggs. And once they lay their clutch of eggs, we would go into the nest, remove the real eggs, and then leave artificial eggs behind, which the adults would then continue to sit on, thinking that they just scared off a predator. They were pretty good replicas, though. But they will just sit on a white rock, too. So we'd then take them back. For most of the period, we were actually flying them up to the San Francisco Zoo, where they were artificially incubated. And then hopefully 5 weeks later, we would get a hatching chick. It's a 35-day incubation period. Hatching actually takes about 2 days. Day one, they'll break into the air cell at the big end of the egg and start breathing air. Next day, they'll actually punch a hole into the eggshell. And then the third day, in about an hour's time, they open up the end of the egg there. So it's a long, long wait. They're not that cute, though, when they come out. little wet and red. But after a couple hours, their down dries out, and they get really cute. So as soon as they've taken their first poop, so that we know their digestive system is working, then we can start feeding them. Starts out with just a gram of usually ground quail, which is easily digestible, but it's got calcium and everything in it. And then by day 2, day 3, they're eating about 50 to 75 grams per meal. So between 1989 and 2004, we removed 81 eggs, seen here in the yellow, of which only 14 hatched, seen in the red. So less than an 18% hatching success. Fortunately, the zoo had 6 to 7 pairs of breeding bald eagles, and we were able to get any of the chicks that they produced to bring down to Catalina and foster into the nests. In 2004, the Montrose trustees who were funding us asked us to try the incubation on Catalina Island. So in our office on Catalina and Avalon, we got an incubator and started incubating the eggs on our own. In my first year, 2005, I hatched 3 of 9 eggs, which was almost double of what they'd done at the zoo. And then by tweaking humidity and temperature a little bit over the next couple of years, I was able to actually hatch 7 of 9 in my third year. And so that was about 78%. So I greatly increased the hatching success. And I think part of it was due to our incubation techniques, and part was due to not having to transport them to San Francisco. That's a long trip for a very fragile. They're only a few cells thick at that point, so that probably is not the best thing for the developing embryo. So these are the 7 from 2007, ranging in age from about a few hours up to a few days of age. These guys eat about every 2 hours throughout the daylight. So imagine feeding 7 birds on a cycle. You pretty much, as soon as you finish, you're starting prep for the next meal. When they're usually about a week old, we then pack them into a carrier to take them to the nest for fostering. On Catalina, a lot of the nests are on rock outcroppings and pinnacles, so we're able to walk or ski or get to them. So these are usually about 300 feet above the water. Most of them have a pretty good drop-off. Other nests were not so easily accessible, and in that case, we had to be a little bit more creative, and we used what we call the dope on the rope technique. So bring over a helicopter, lay out a 100-foot rope, attach it to a harness, which is attached to the bottom of the helicopter, and take a little ride. Yeah. I've probably done maybe 40 to 50 of these. Fortunately, still I am, really. So I had a very good pilot, though. He puts air conditioners on buildings and stuff like that, so he's very precise. First one, that clip actually came loose because it had been dragging on the ground while we were doing stuff. So I got up on my toes and it let loose. So I just grabbed it and hooked it back on, and probably didn't look down much that first few times though. So then this is probably about a 5-minute ride to the nest. The reason the rope is so long is so that the propeller downwash doesn't blow the nest too much. So we just go in, switch out the eggs, put in the chicks. Yeah, we're scaring them. Yeah, they're not real bright. These are about a week. Yeah, we'll see the adult actually come back, and this is in real time, so this is how quickly the adults can come back to the nest. Not necessarily, especially because so many didn't hatch. Most of these are chicks from San Francisco. Of what? That particular one, it's either 65. I'm not sure if that's the male or the female. So once the chicks were in the nest, we'd watch the nest 2 or 3 times a week to make sure that they were doing well, being fed. Depending on the nest, we can either set up a blind and watch them through a spotting scope, or set up a remote camera. Some of these nests, they're just not in a spot where you could actually see into the nest through a spotting scope. So I'm sure a lot of you are familiar with our live webcams. The West End and Two Harbors nests, there's no way to actually look into those nests from anywhere, so that's why we've set up poles next to them and put cameras on so that we can actually see what's going on. When the chicks are about 8 weeks old, we then go back. At this point, they're not able to fly, but they're essentially full grown. So we remove the chicks temporarily for banding. On Catalina, they get an orange wing marker, which allows us and others to identify them. They get a temporary radio transmitter backpack, which we're able to go out with a receiver and antenna and track them around. But those are designed to fall off maybe after 2 or 3 years. They're just, they're like a backpack, 4 straps that are sewn together with thread, and that thread just breaks down. It's embroidery thread, and it breaks down, and then the backpack falls off. Because the battery life is only about 2 years, and we don't want them carrying around all this extra weight, although they're pretty light when they're not functioning. And they also get a leg band. We draw a little bit of blood to look at their contaminants, and then put them back up in the nest. In 2005, 2002, sorry, we began a similar project up here on Santa Cruz Island. There was a lawsuit settlement against Montrose Chemical, which provided a lot of money for restoration work, and we were asked to try releasing eagles on Santa Cruz because it was further from the contamination source, and at that point it really didn't look like eagles on Catalina were going to be able to hatch anytime soon. So we essentially doubled our effort, releasing 61 eagles over a 5-year period. We'd done 33 eagles over a 6-year period on Catalina. So we put out a lot more eagles at one time, hopefully to get a bigger population going. Used a similar technique with the hacking tower. Up here, they got blue wing markers so that we could tell the eagles apart between the different projects. They also got a GPS transmitter backpack. So now we didn't have to physically go out and look for the birds. We were able to actually just go on the computer, download some data, and up to 14 times a day we would know where each bird had been. So this is just a small representation, but they move easily among the islands up here. Sometimes an eagle will be on all 4 islands in a single day, so it's just impossible for us to physically go out and track them. They also, a lot of the young birds tend to go to the mainland and travel around, but mainly in the Pacific coastal area, but some head over to Montana, Nevada. We've had them up into central British Columbia, and then they'll come back down, usually to Southern California. These also gave us a good insight into probably the major mortality cause of eagles on the islands, at least, and that's drowning or hypothermia. So, it was a sad day when we'd download the data and then you'd start getting, this kind of thing floating out in the water. It gave us a good look at the currents of Southern California, but so that's probably the major cause of mortality for eagles out on the islands. And we don't know if they tire out trying to fly either between islands or to the mainland, if gulls or some other bird sort of forces them in the water, if they try to land on the water not knowing it's not solid, try to land on a kelp patty. We just really don't know. In 2005, we got our first apparent pair formation. Ironically, it was actually birds from Catalina Island. The male had been fostered into our Twin Rocks nest on Catalina in 2001, and the female our West End nest in 2002. And then they found each other up on Santa Cruz Island. In 2006, we finally got our first breeding, about when we expected it to happen. They generally don't breed until they're about 5 years old. So we had 2 nests on Santa Cruz Island in 2006. The Pelican Harbor nest was those 2 birds from Catalina Island. They hatched the first chick known to hatch on the island since 1950, and it was a pretty exciting day for everybody. This bird is actually still alive, and you'll see her again in a little bit. And then we had our Malva Real on the south side of Santa Cruz Island, which was a very strange nest. They actually just put some sticks and grass down in a field and laid eggs there, and they actually hatched 1 chick. It survived, and it's also still on the islands. So in 2007 on Catalina, because I'd been having that increased hatching success in our incubation, we'd had that successful breeding up on Santa Cruz Island, we decided to leave eggs in a couple of the nests down there. And then we continued to remove the eggs and incubate them for 3 other nests. So we left 2 eggs in what we call our Pinnacle Rock nest on the south side of Catalina. They both hatched and both fledged. And 2 eggs in our Seal Rocks nest also hatched and fledged. So that was, in 2005, I didn't think I was ever going to live to see eagles hatch on their own on Catalina. So this was very exciting for everybody. Let's see. Yeah. And then 2008, we were building on that, so we left eggs in our next nests. The reason we left them in the Pinnacle Rock nest was the female was the youngest bird on the island, so we thought maybe her contaminant load would be low. And Seal Rocks, our measurements of the contaminants had been lowest in that territory for 5 or 6 years. So again, Pinnacle Rock successfully hatched 2 chicks. Seal Rocks hatched 1. What we call our rattlesnake territory here, they laid eggs, but they failed to hatch. Then we left eggs in our Twin Rocks territory up here, and they too successfully hatched 2 chicks. We'd actually taken the eggs out of the West End nest here, but we only took 2, and apparently they laid a 3rd one after we'd done the egg switch. So the day before we were going to foster chicks back into the nest, this little guy popped up on the webcam. So they actually had successfully hatched a chick, and that's the nest that we considered our most contaminated. So in 2009, we figured, we'll just leave all the eggs. What we thought was the most contaminated is hatching eggs, so we'll leave them all. Again, Pinnacle Rock was successful with a chick. Seal Rocks with a chick. The rattlesnake ones, again, laid eggs but failed. Twin Rocks, again, had 2 chicks. West End had 2. And 2 Harbors had 2. So this was the last nest that we'd left eggs in. So everybody appeared to be having pretty good luck. We also had our first mixed pair formation. So we had a female that came down from Santa Cruz Island to Catalina and paired up with a male from Catalina, but they didn't lay. They built a nest in a eucalyptus tree near our reservoir, but they did not lay eggs. So in 2007, we had 7 nesting attempts on the island, and again, Pinnacle Rock, they've been successful every year since we'd left the eggs. Seal Rocks again. Rattlesnake finally had their first chicks. So that was added to the number of successful breeders on the island. Twin Rocks had eggs, but they didn't hatch. And they actually sat on those eggs for, I think, close to 11 weeks. And it's a 5-week incubation period, so they don't give up very easily. Then the West End had twins again, and 2 Harbors had twins. Those of you familiar with the Ojai Raptor Center, Avalon, she went up to the Pacific Northwest and broke her wing, so she's now an education bird down in Ojai. And then the Middle Ranch pair had their first chick. So at this time, all the birds that we'd left eggs with had hatched at least 1 chick. 2011, again, we just had the 7 nests. This was the first year that Pinnacle Rock did not successfully hatch any eggs. Seal Rocks had 2. Rattlesnake again had 2. Twin Rocks failed again. The West End had their first set of triplets. And 2 Harbors had a single chick. Sassy. Middle Ranch also hatched a chick, but it died within about a week. So I have no photos of that one. And then finally, this year, we had 6 nesting attempts. You'll notice that Twin Rocks is not on here. There's a new male this year, and they didn't even attempt nesting. So Seal Rocks, again, had 2 chicks. Rattlesnake had 1. Well, actually, they had 2 chicks, but 1 disappeared during a windstorm, and we were unable to find it. It was probably about 3 weeks old at the time, so I assume an island fox probably ran off with it. West End, again, had triplets this year. Those of you watching, it was a pretty rough year for a lot of our birds. I think there was a lack of fish in the environment during critical period. So this chick in the front was quite the runt when we went in to band it. So she didn't get wing markers or a radio transmitter. And then Two Harbors, this was probably the saddest one of the year. They hatched a chick, and I think because of a lack of food, the adults left it alone overnight when it was about 3 weeks old, and an island fox actually came into the nest and walked off with it. So they're not supposed to leave them alone at that age. And I just think they're out looking for food, and they just can't find food before it's time to go back to the nest. So, we've been holding steady at about 5 to 7 nests per year on Catalina. Up on the northern Channel Islands, we're really starting to take off. In 2007, again, we just had those first 2 nests that we'd had successes at in 2006. Pelican Harbor, again, hatched a chick and fledged. And these guys tried nesting on the ground again, but their eggs did not hatch. 2008, we doubled the number of active nests, including adding our first nest over on Santa Rosa Island. Pelican Harbor, again, had 2 chicks at about 7 weeks of age. These guys were knocked out of the nest by a subadult bald eagle. One of them had a broken wing, and one had a puncture hole through its beak. So they were rehabbed and then released from one of those hacking towers, and one of them is still alive, and you'll see him again in a minute. The Malva Real pair in the grassland hatched 2 chicks, but they died a couple of weeks old. We think the nest was attacked by another adult female bald eagle. One of the chicks was found dead, and the adult nesting female was found dead a couple days later. So somebody wanted that territory. And then we added the Sauce's nest down at the west end of Santa Cruz Island. This is one of the ones that we have live online now. They had eggs, but they did not successfully hatch. And then we have what we call our Trap Canyon pair out on Santa Rosa Island, who began nesting in an old golden eagle nest. But again, their eggs did not hatch. 2009, we dropped back down to 3 nesting territories. The one where the female was killed, he did not really have a mate that year, so they didn't attempt breeding. It was another sad year for the Northern Islands. The Pelican Harbor pair had 2 chicks, each of which died at about 3 days of age. Really don't know why. It could have been domoic acid poisoning. There were some outbreaks at about that time. So when they're that small, that could really affect their health. And then the other 2 nests, we had eggs but no successful chicks. In 2010, we added another nest on Santa Cruz and down on Santa Rosa. We had Pelican Harbor was finally successful again with 2 chicks. Sauces had their first chick successfully. We added the Cueva Valdez nest up on the north coast of Santa Cruz, and they had a chick. And then we had the first chick for the Trap Canyon pair. They moved out of that golden eagle nest down closer to the water. That golden eagle nest was about, I think, 4 kilometers from the coastline. So even if they'd had chicks, that was going to be a long distance to have to carry fish several times a day. So they moved down. They had a very nice view of the ocean. And that was the coolest nest I'd been in to that point, because it was under an overhang. So they were out of the wind and the rain and the sun and very well protected. And then we added the Lopez Canyon nest on the south side of Rosa, which also successfully raised a chick. In 2011, we increased to another island, Anacapa Island. Pelican Harbor, the previous nest they were in was just disintegrating, the tree was dying, the nest was falling apart. So they tricked us this year. They started building a nest in January, so we went in, put a quick camera on it so that we could watch it, and then they moved about 5 miles down the coast and built another nest. Yeah, they were tired of being on TV. Sauces again had a chick. Cueva Valdez also moved their nest several miles down the coast to a new location and had a chick. Then over on West Anacapa Island, we have what we call our Oak Canyon nest. Another researcher actually found this while they were out doing some vegetation monitoring, I believe. He'd been one of our previous employees, sent us an email saying, I found this nest, here's where it is. West Anacapa is very inaccessible during the spring because of seabirds. So basically in June, we just took everything that we needed to band, hoping that there was going to be something there to band and of the right age. And when we got to the nest, there was a perfectly aged eaglet in there, so that one got banded. Over on Santa Rosa, both of those pairs had laid eggs. This was when they were working on the pier, so it was sort of difficult to get out to the island. So we were only really going out about once a month. So when we went back out in early April, both of the nests had failed. Although we did find a dead chick, probably a week or so old, under one nest. So it looks like it had hatched at least one chick. It just, something happened. And then finally this year, we really took off with 10 breeding attempts on the islands. And you can see we're starting to fill in the coastline of Santa Cruz Island. I hope to start seeing that similar sort of thing out on Santa Rosa where there's appropriate habitat. So 10 nesting attempts. Pelican Harbor left the nest they had the previous year where we set up this very expensive camera system. Went back to the nest where we'd set up a little cheapo bullet camera the previous year. So we at least had a video on them. Malva Real got a new female, possibly a new male as well, and they had 2 chicks. This one's in a pine tree miles inland, and it's very hard to see. And when I was traveling out to band, I got a call that one of the chicks was missing. They only saw one chick in the nest, so we got there, spent probably half an hour or so just going back and forth across the hillside, and I found the other chick just standing on the ground about 100 yards up the hill. So we were able to band it, put it back in the nest. It probably blew out during a windstorm as well. So they had 2 chicks. Sauces had 2 chicks this year, and they're both still alive. One of them, there's some really bad erosion on that portion of the island, and one of these birds had gotten down into an erosion gully that had no ends to it. It was just a crack in the ground, probably 10 feet deep and 3 feet wide, and somehow it had gotten down into there, and it couldn't get back out because it couldn't flap, it couldn't extend its wings. So fortunately, because of those radio transmitters, one of my assistants was able to find it and get it out of the hole in the ground. So it's doing well now, but it would have been dead without that radio transmitter. Cueva Valdez, they had a chick. We banded it. And then a couple weeks later, it was drooping its head for some reason. And I was up in Washington on vacation, so we really couldn't get into the nest. A day or two later, they went back to check on it, and it was on the ground and really not moving, but it was still alive. So they took it to the Ojai Raptor Center, ran all sorts of tests on it, couldn't find out what was wrong, and it did die a few days later. But that's very rare for the eagles to die between the time we banned them and the time they fledged. So this nest, it's got very low branch cover overhead, so it could have been bouncing around, exercising, and just whacked itself good on the head or injured its neck. I'm not really sure. We had another new nest, our Fraser Point nest, and that's where I was bringing back those Pelican Harbor birds. A49, the first chick to hatch on the island. They built their nest in a pretty poor location, though. It's up near the ridge top, probably about 1,300 feet. Most exposed, windiest area you could probably select on the island. And she also paired up with her brother from 2008. So we're pretty sure they had a chick. We think we saw a chick moving around, but it's a very difficult nest to look into. But it disappeared within a week or so. So this was their first breeding attempt by an eagle that actually hatched on the islands. And we hope they choose a better nesting location next year. It's not uncommon for birds to be unsuccessful the first year or two of breeding. You know, they might not know how to incubate, leave the eggs alone, step on them. But and with the chicks, the first nest in 2006, they didn't feed the chick for 3 days. And, you know, it was the first chick to ever hatch. The park was putting out a press release. And, you know, I was getting on the boat to leave the island to go back to Catalina. And it's like, you know, we're putting out this press release right when the bird's going to die. But now we know they can go for at least 3 days without eating. Because when they hatch, they reabsorb the yolk in the egg. So they do have that energy source. So now I'm not so concerned about them not eating for the first day or two. Yeah. Then we had 2 other first-time nests this year. We had Fry's Harbor. One of these is A46, also known as Stephen Jr. He was named after Stephen Colbert. So he's been featured on the Colbert Report a few times. And then we had what we're calling the Los Pinos pair. They also laid eggs. Both of these, their eggs did not hatch. Anacapa didn't hatch this year as far as we know. Again, it's a very difficult nest to watch. But they definitely did not raise a chick. And then on Santa Rosa, Trap Canyon moved their nest again because I'd put a camera in the previous one. So this one, the only place you can see into the nest is from right there, and that's about 50 meters away. The adults really don't like that. So I was out there last weekend. I've moved the camera stuff from the previous nest over here, and hopefully we'll be able to drop a camera off the edge of the cliff from above and be able to watch them from up the gully without the adults seeing us. Of course, they'll move back to the nest I just took the camera off of. And then the Lopez Canyon had another chick this year. They actually had 2. Again, one fell out of the nest early on. We found it underneath the nest. So I don't know why they keep, maybe one of the birds is a bully and keeps knocking them out. So just as a summary, we've had 234 total successful fledges during this project, including from the hacking towers in the red, from fostering chicks into nests in the green, and now from natural reproduction in the blue. And believe me, having the natural reproduction is a lot easier for everyone involved. It's pretty stressful to have eggs and chicks in your office. And the number of active nests on the islands are, you know, they were holding steady for the first 10 to 20 years. Now they're really starting to take off. I think we're going to hit 20 in the next year or two. And I'm really hoping we'll have 20 chicks produced, possibly next year if all 8 of those Santa Cruz ones breed successfully and we get all our Catalina birds breeding again. So I think we could easily hit 20 chicks successfully fledging next year. So right now we estimate that there's about 20 to 25 eagles on Catalina. That number varies. Young birds come and go from the islands at all times. I've actually seen an adult fly from Catalina off to San Clemente Island when I was on the coast. So they move between the islands. They're looking for mates. Up on the northern islands, we think there's about 30 to 35 eagles at this time. And it's possible that we missed a nest on San Clemente Island this year. We think there might be a nest down on the south side. That's another hard island to access. This is the area where they bomb, so there's a lot of unexploded ordnance, and we're not really allowed in that area much, but I think we're getting permission to get in there for next year. So it's possible that we have a breeding pair there. They did see a young eagle this year. I couldn't say from the description of it how old it was, so it could have been a 1 or 2 year old boy. It didn't have any wing markers on it, so Where? Yeah, that you mean a few months ago, or yeah. But this one, the bird that they saw, didn't have any wing markers or anything on it. Yeah, real well. So we estimate there's about 60 bald eagles on the islands right now. Expect this to increase as we started getting more and more natural reproduction. I think more of the eagles stay on the islands when you, one, have lived your whole life there, hatched and raised there, and two, when you have a lot of younger birds to hang out with. They sort of follow each other around and find food sources. They're primarily scavengers for the first year, so you'll get a bunch of young eagles feeding on each other, or not each other, hopefully. I've not had that yet. Feeding on, especially on the northern islands, dead marine mammals. Until this year, deer and elk carcasses. There were deer and elk on Santa Rosa, but those have been removed. So I expect to see the birds along the coastlines more now. The sea mammal populations are really starting to take off as well. So there should be plenty of food out there. And with that, I'd just like to thank this long list of people that have helped us conduct this research. Right now, we're being funded primarily by the Montrose Settlements Restoration Program, which oversees the money from that lawsuit settlement, but couldn't have done the work without all these people. Thank you, and I'd be happy to answer any questions. Yes?
How long do bald eagles live, and how long are they sexually active?
They live, the oldest record in the wild is about 35. They can live 40 plus in captivity, and they're active until they die, essentially. So starting at 4 to 5 years until they die.
The eagle that was found over on Palos Verdes, any more word on?
We've had no more sightings of that one. One of the West End chicks was over by the Trump golf course on Palos Verdes, so it successfully made it across the channel, but we haven't had any more sightings.
Doesn't have a transmitter on it?
They have VHF, which means we physically have to go out and track them, so we have had no signal from Catalina.
I saw a blue-tagged one on Gaviota Coast. Are we supposed to report these anytime?
Most likely that one has a GPS transmitter on it, but feel free to email me with sightings.
Okay, are you going to have the telescopes on the Catalina Marathon on? You think there's some nests there this time again?
If they nest, I'm hoping to have a live camera on that one that's fed back to the Eagle aviary. So that's what we'd do.
Would you anticipate them moving out to San Nicolas and Santa Barbara Island at some point?
Eventually, yes. They nested on all of the islands. I think they're going to probably take up the space on the larger, closer to the mainland islands first, and then move out to Miguel and Nicolas and Santa Barbara.
What do you think the carrying capacity of the islands is?
Well, the most that we're known to be breeding is 25 pairs in a single year. So that was the estimate, but I think that's probably quite low. I would think, we could probably have 10 pairs on Catalina, Santa Cruz, Santa Rosa, 2 or 3 on Anacapa, probably 2 or 3 on San Miguel, 1 on Santa Barbara. So we could probably be in the 40, 50 range. That's pairs.
You must have hundreds of people all over the world watching these eagle cams and developing clubs and raising money and T-shirts and what's been the impact on your work?
Just a lot more public outreach. Just seeing how many people are involved and interested in the work that we're doing. So. I think I put the first cam online in probably 2005, maybe 2004. Had a small group that watched those. And then as technology has increased, we've been able to those first ones, I think we had an image every 5 seconds or something. And then, we got streaming cameras, and then now we've got pan, tilt, zoom cameras on nests. And you know, it's a lot of answering questions, and it's getting people involved and interested in what we're doing.
What are the current DDE levels being measured?
Well, we're not really measuring anymore because what we were measuring was in the eggs, and we don't get eggs anymore. We haven't even done the blood recently just because it really doesn't tell you a lot. It's pretty low in the chicks at 8 weeks of age, but they do have it. But it doesn't really tell you much because they've only been feeding for 8 weeks in the environment. So we have occasionally tried to recapture older ones to draw blood samples, and it is going up. And as they get older, they pick up more and more. So right now, although you know everybody's hatching their eggs, one of our concerns is that potentially they could get to a point as they get older where they've picked up too much DDE and again can't hatch their eggs. So that's for the next 5 years. We're going to be monitoring the nests and make sure that something like that doesn't happen.
They have any type of predator?
Do they have a predator?
Yes.
The adults don't really. The chicks, when they're young, they can be taken by gulls. They can be taken by ravens. We've had one taken by a red-tailed hawk, just right off the nest with an adult eagle standing there. Foxes. But for the adult flying eagles, humans are really their only predator.
The question I have, since you started in about 1980 on Catalina, how old are the, let's say, 5 oldest bald eagles you've got?
We've got one female from 1984. Ray on the west end is from 86. And I think most of the other ones are quite a bit younger now. So in the last few years, we seem to have been, our older birds have been disappearing. So those are our 2 oldest ones.
Are you taking blood samples from them to see what the DDT levels are?
No, I guess we're sort of assuming that if they're hatching chicks, then we don't really need to be so concerned about their DDE levels.
Is the success rate, or over here, is the success rate that we're seeing on the islands typical of bald eagles elsewhere, or is it lower? And if so, why?
It's similar to, and I think our survival of the young birds is actually higher than mainland for the most part. Mainland, it's generally about 50% survival through the end of the first year, and we're more in the 60 to 70% usually. Many years, especially on the northern islands, we'll have 100% survival. So as long as they're not flying across large expanses of water and not going to the mainland, they have pretty good survival.
You mentioned, it's on right. You mentioned domoic acid as a potential for, in one instance. You thought, is there any trends or you're seeing there? We know with the shorebirds, I was wondering if it's affecting the eagles at all.
The domoic acid, it's really hard to say. I don't think it would affect the adults as much. I just don't think they'd be getting a lot. I guess I'd be a little concerned if they were scavenging on like a dead sea lion that had succumbed to domoic acid. You know, maybe there'd be a chance of picking up a larger dose. But I think the big threat would be mostly to the very young chicks, because they only weigh about 90 grams when they hatch.
Peter, do you know if the California condors that tried to hatch a couple of years ago at Big Sur and failed because of DDE in their eggs, they tried again?
I haven't heard whether those birds tried again or not, but I know DDE is a concern now for them. You know, things feed down here and then move up the coast, the sea lions and everything. So the condors are feeding on washed up sea lions or something up there and picking up the DDE. So it spreads from down here in animals' bodies up to different areas and can cause problems up there. Because they have the sea lions and elephant seals have a very large foraging area.
You mentioned temperature and humidity changes raised your success rate on the hatching. What do you think, or what have you found is the optimal conditions to hatch these guys?
Well, we were using a different kind of incubator. Generally, I don't have the controls here. Generally, eggs are in what's called a forced air incubator. You just put them in. It's got warm air that moves all around, and those are usually about 99.5 degrees, just warm air all around. What we used was called a contact incubator. So the eggs are put down on a roller, and a balloon essentially inflates and lays down over the top of them like an adult bird would. That was at 102 degrees, which is about the body temperature of an eagle. About every 45 minutes, the balloon would deflate, the eggs would roll, and then the balloon would reinflate, and that's the adults roll the eggs about once an hour so that the membranes don't stick together. And then I just increased the humidity as much as I could, because our biggest problem with these eggs is that they lose water too rapidly, because the eggshells are a little bit thinner, and they also have larger pores, so they just lose water. When I was taking them out of the nests, some of the nests, they're supposed to lose about 15% of their weight and water loss over the 35 days, and some were doing that in the 2 to 3 days that they were in the nest before I took them. So just by the typical belief is that you keep humidity low in an incubation room, just I guess to keep it clean so that molds and stuff aren't growing. I eventually just brought in a humidifier and increased it to 40 or 50% in the room, and the weight loss in the eggs was able to trend without. What we would do with the ones that had really high water loss was we'd put tegaderm on it, which is used for burn victims. It allows gases to move in and out, but not moisture. So we'd cover portions of the egg with that to keep the weight loss down to where it should be. So by just increasing the humidity in the room, I was able to minimize the need for that tegaderm. So 102 degrees and 50% humidity.
Here's sort of an evolutionary question. You mentioned that the success of bald eagles generally on the mainland, in a sense, is 50%, and it's much higher on the islands where there may be historically more ideal conditions. The fish are there. There's less predators. Do you think that historically the island bald eagles were actually supplying the mainland more than vice versa?
Definitely, I would think so, especially in Southern California. Before we put in reservoirs and everything, there was really only the coastal habitat for bald eagles. So I would think we were probably a source for helping with the Big Sur population. If there's a record in the late 1800s, somebody saw 30 eagles soaring over the western end of Catalina, 30 young eagles. So, those were probably produced in over a year or two. And the West End, for some reason, all the birds end up out at the West End. I think they're trying to just go north and they run out of land. But you'll find just about every bird that starts flying on the island will eventually make their way out there. So, I think we probably were a source for a lot of the areas around here.
Well, thank you, Peter, for a very interesting talk.